Greetings everyone, we as a collective are members of Group 4 and today we'll be presenting about one of the direct plant transformation approaches, which is microinjection based on the paper Shoot Apical Meristem Injection, a novel and efficient method to obtain transformed cucumber plants by Bhaskaran et al. Here is the list of our group members. Without further ado, let's begin. Cucumis sativus L is one of the important vegetable crops and had received recognition as a model plant for research in its family. However, the cucumber's yield is severely affected by phytopathogenic microorganisms and it is a problem faced worldwide. Luckily, recombinant DNA technology serves as a novel way of improving disease resistance in plants whilst minimizing the loss and is an alternative to conventional breeding method. Gene transfer by agrobacterium tumefaciens is normally used in this case, but it is rather uncommon to be used on recalcitrant plants like C. sativus due to the lack of efficient transformation and a regeneration system. So, Bhaskaran et al. Had done a research to develop rapid cucumber transformation through microinjection on shoot apical meristem or SAM. You might be wondering, why SAM? Firstly, it is easy to culture in vitro. Second, it is known for its rapid regeneration. Third, it is more competent to genetic transformation and also produces genetically identical plants. Lastly, it sustains for much longer time in vitro. The ultimate objective of the study is to study the efficiency of microinjection in transferring genetic materials to the shoot apical meristem of cucumber using shooter strain of Agrobacterium.
In the beginning, the seed of the cucumber was soaked in water and dishusked manually. Later, the seed proceeded to the decontamination step. After decontamination, the seed were inoculated in the germination medium. The pH of the medium was adjusted to 5.8. All the media are autoclaved under specific conditions. The seeds were left alone in the dark. 3 days later, shoot apical meristem SAM explant were excised from the germinated seedling for the subsequent experiments. Triparental mating is a way of bacterial gene transfer involving 3 bacterial strains. The first strain contains conjugated plasmid that promote the transfer of mobilizable plasmid from the second strain into the third strain. So, by this gene transfer method, the binary vector in one strain was successfully transferred into the shooter G strain with the help of the helper vector in another strain. This binary vector contains MPT2 and NSCP genes, which are responsible for kanamycin and cucumber mosaic virus resistance, respectively. The resulting strain called shooter G and SCP were then cultured in YEB medium, leaving it in the dark until an optical density at 600 nanometer of 0.5 to 0.6. Later, centrifugation was performed to get the pellet of the bacteria. The resulting pellets were resuspended in the microinjection medium and shaken for a few minutes. This bacteria culture were all set for a microinjection of SAM and infection of the cotyledon explants.
Secondly, let's look about gene transfer by microinjections. In this article, we will find out that there are 2 microinjections that have been done. Firstly, pre, for the testing purpose, and second one will be the actual microinjections. Now, let's look about pre-microinjections. The bacteria was microinjected 3 times into SAM region of shoot tip explant from 3-day-old in vitro germinated seedling using a needle followed by blotting the shoot tips on filter paper. Next, the microinjected SAM of the shoot tips was co-cultivated on MS medium for 2 days in the dark. The explant were transferred to different bacterial eliminations liquid medium for different elimination periods, followed by transfer to bacterial elimination solid medium to standardize the K4-testimine concentrations and period needed for eliminations of excess bacterial growth. Finally, the shoot of 1 week old in vitro seedling were collected and transferred to MS medium. Before the actual gene transfer procedure takes place, Optimizations and testing of various factors were done beforehand. That's all for the pre-microinjections. Now, let's take a chance to look at the actual microinjections. Microinjection on 3-day-old SAM were conducted from optimizers, culture media, and conditions. The culture will refresh every 3 weeks for 6 weeks. No exogenous growth regulator will apply in the experiment. The shoot was transferred to optimized selection medium. The culture was maintained for 6 weeks. Then, KM-resistant rooted plants were separated and washed in sterile distilled water and then plotted on filter paper. Finally, the plant was transferred to a greenhouse. Now, 3-day-old cotyledon explants were removed from germination medium and were immersed in infection medium 1 containing bacterial suspensions for 3 minutes on a shaker at 80 rpm. This purposely to bottle off the excess bacteria by filter paper. Next, the explant were then transferred onto cold cultivation medium for 2 days in the dark. The selection will be done and the transgenic plantlets are grown healthily.
All experiments were repeated 3 times with 50 explants per treatment. The data collected were calculated using ANOVA and Duncan multiple range tests. Before performing PCR analysis, the DNA of the cucumbers has to be extracted. Later, PCR was performed in 3 replicates according to the standard conditions. As a result, the status of the transgenic plants can be confirmed.
As mentioned in the protocol and materials, optimization of culture media and conditions were done to test a suitable environment for microinjected shoot apical meristem of cucumber explants to be cultured in the actual experiment. Optimization relies on the type of antibiotic, explant, cultivar use, and concentration of the reagents. It was determined that the concentration of antibiotic cefotaxime that would be added to liquid medium and sodium medium to have 0% bacterial growth was optimal at 300 mg per liter with an elimination period of 10 minutes. Similar concentration of cefotaxime was also tested with a higher elimination period. However, the plant will produce hyperhydric shoots at 30 minutes, while it also shows signs of hyperhydric shoots with growth of white compact basal colors at 60 minutes mark. Transgenic cucumbers are resistant to kanamycin. Kanamycin inhibits root growth, while shoots deteriorated drastically with time. The shoots of 1 week old in vitro seedling generated from disinfected seeds were collected and transferred to MS medium containing glucose, agar, indole acetic acid, indole butyric acid, or alpha-naphthalene acetic acid, and Km to standardize the auxin and Km concentration for rooting and selection of putative transform shoots. This is the investigated minimal inhibitory concentration for selection of transgenic Km-resistant cucumber. It was found that the concentration of kanamycin of 25 mg per liter is the best minimal inhibitory concentration for the selection of transgenic cucumber plants. At this concentration, it's just enough to inhibit root growth and cause shoot necrosis of non-transgenic plants that have a lack of kanamycin-resistant gene. Among different infection conditions tested for efficient gene transfer, it was reported that the germination and infection condition of infection medium 2 plus acetylsyringone with agitation coupled with the germination medium plus 100 micromolar acetylsyringone show the greatest efficiency among others, as approximately 65% of KM-resistant rooted plants were grown. This condition allows the bacteria to penetrate into the cucumber shoot meristem, including the germ lines, and subsequently facilitate the gene transfer. The microinjection treatment with shoot apical meristem allowed successful transfer and incorporation of tDNA into the cell, possibly by inducing production of gene-inducing compound. After 6 weeks of culture, the efficiency of gene transfer was tested through physical observation. The cucumber explants that were microinjected with bacteria through shoot apical meristem had produced 4 with ±0.45 shoots per explant with Km-resistant gene transfer efficiency rate of 65%. In the case of cotyledons that were immersed in bacterial suspension, only 3.2 with ±0.45 shoots per explant were observed, which is significantly lower than shoot apical meristem explants that were microinjected. Moreover, only 35% of them were identified as Km-resistant shoots, which results in a 43.8% gene transfer efficiency to be recorded. However, the control non-infected plant did not grow any shoots as it did not survive in the culture due to not being resistant by kanamycin. The plantlets generated were transferred to vermiculite and sand with the mixture in the ratio of 1:1 and then maintained in the growth room and greenhouse. After PCR tests for the transformation of the cucumber plants, the transgene is confirmed using DNA through electrophoresis for both putative transgenic plants and in vitro transgenic plants. As shown in the diagram, the DNA of the shoot apical meristem transformed plants and cotyledon transformed plants were compared with the DNA ladder at 660 base pair marker representing MPT2 gene. The similar migration distance from the electrophoresis result proves that the gene of interest, MPT2, has been successfully incorporated into the genome of the transgenic plants. However, the thickness of the bands between Agrobacterium transformed cotyledon and Agrobacterium microinjected shoot apical meristem differ, as shoot apical meristem showcase a thicker band than cotyledon, which correlates to the efficiency of gene transfer, where thicker bands means higher DNA concentration. Shoot apical meristem has a higher efficiency in being transformed with Km-resistant genes.
In conclusion, a high-efficiency cucumber gene transfer by SAM microinjection using Agrobacterium shooter strain has been successfully established. Because of the careful selection of explants, it minimizes the chances of obtaining false positive and chimeric plants. By using PCR, the gene of interest which has been incorporated into the plant was confirmed. As the technique develops, it possibly can provide us with ways to study the cucumber and pave the way to molecular breeding to breed crops with a wide variety of traits. That marks the end of the presentation. Thank you for watching.